Hello everyone, today we're going to talk about the synaptic transmission. As we know that the step of transmission of the nervous system at the level of the synapse can be shown as the following. We can make it as steps. First, the action potential arrives at the axon terminal. You see this right here? This is called the presynaptic neuron. This is called the postsynaptic neuron, and this right here is called the synaptic cleft. The synaptic cleft is the part or is the gap that is located between the presynaptic and the postsynaptic. The presynaptic neuron is the one that translates the message from right here into the postsynaptic neuron where it can continue. First, the action potential or the nerve impulse arrives to the end of the presynaptic element or the presynaptic neuron. You see these right here? These are voltage-gated Ca2 plus channels. When the action potential arrives, these channels will open and Ca2 plus will step in to the presynaptic neuron. So they open at the level of presynaptic element, which leads to the entrance of calcium ions inside the terminal bud. After that, there is liberation of the neurotransmitter by exocytosis into the synaptic cleft. Let me explain. When the Ca2 plus enters to the presynaptic neuron, these blue stuff, they are called synaptic vesicles. They are like bags that contain these pink dots. These pink dots are called neurotransmitters. When the calcium enters, these synaptic vesicles will bind to this membrane right here, and the neurotransmitter will be released into the synaptic cleft. After that, there is the release of neurotransmitter. This is the exocytosis. So when the neurotransmitter is released from the presynaptic to the synaptic cleft, this step is called the exocytosis. So there's a liberation of neurotransmitter by exocytosis into the synaptic cleft. After that, these neurotransmitters bind to these chemical receptors. These chemical receptors are located on the membrane of the postsynaptic neuron. They are ion channel receptors, so they are chemical. They do not depend on voltage like the voltage-gated Ca2 plus channel. This neurotransmitter binds onto the receptor, which causes it to open. And after that, there will be here in the synaptic cleft, there will be Na plus or K plus. They will go into the postsynaptic neuron, which will generate an action potential right here. Well, that is in case the synapse is excitatory. If Na plus goes in, this will lead to the creation of exocytatory postsynaptic potential, EPSP, that could generate AP if it exceeds the threshold of excitation. And in case if it's an inhibitory synapse, the channels that open are Cl- channels, and this will lead to an inhibitory postsynaptic potential, IPSP. So for a quick summary, if Na plus enters, this means that it's an excitatory. If Cl- enters, this means that it's an inhibitory synapse. And if you want to know in another way, you can see right here in the postsynaptic. If there's a generation of action potential, then it's an exocytatory synapse. And if it's an inhibitory synapse, there will be no genesis of AP right here. You may ask, what happens to the neurotransmitter that is located on the receptor? This neurotransmitter is destroyed by a specific enzyme and recaptured by a specific pump present on the presynaptic neuron. So the Na plus channels or Cl- channels, they will eventually close.